Hello everyone, welcome to your third lesson in chapter 21, amino acids, proteins, polypeptides. We're beginning with section 21.8, protein hydrolysis and denaturation. When you think of the term hydrolysis, I wonder by now for sure you're thinking adding water, correct? To hydrolyze means to add water. Remember that when we took amino acids and built larger peptide chains, we removed water. So what happens if we add the water back in? Well, you've got it at the exact opposite process. Adding water breaks the peptide bonds and releases the individual building blocks of those amino acids. So this really is a decomposition reaction, isn't it? To decompose back into the original amino acids. By breaking the peptide bonds, you're going to convert the polypeptide chain into individual amino acids in their Zwitter-ion form. And this hydrolysis can be accomplished by adding aqueous acids, base, or even certain enzymes in our bodies can break apart amino acids through the process of digestion. Oops. There, got it. So in our body, for example, there's an enzyme called pepsin. It's found in the gastric juice of our stomach. Pepsin is responsible for breaking down peptide bonds of large proteins that we ingested through our diet, and it begins to break them into smaller peptide chains. Once that has passed out of the stomach and enters into the intestines, there lies trypsin and chymotrypsin in our intestines that begin to hydrolyze the remainder of the amide bonds, resulting in individual amino acids. And of course, those were the building blocks. So this whole process of breaking back down, known as hydrolysis, is a decomposition reaction. Instead of building proteins through dehydration, where we formed the amide bonds, we are hydrolyzing, which breaks the bonds, and we're going to form back our original amino acids. So let's say, for example, we wanted to draw the tripeptide with the abbreviation ILE. And you do this with me. Draw the amino acid that has INE. Let me just get a nice clear slide. There we have it. So now we're going to draw ILE, and it's going to be attached to GLY, which is going to be attached to PHE. How are you doing with your coding? Can you picture what this amino acid will look like? So for example, you know that the N-terminal amino acid has the abbreviation of ILE. The C-terminal amino acid will have the abbreviation PHE. What do those stand for? We need to find what R groups they represent. So you know that as you're working to memorize, these are your amino acid building blocks. The isoleucine, ILE. That's your first of the amino acids that you're building, isn't it? There's the functional group that you need to know represents isoleucine, which has the abbreviation code of ILE. What about the next one? GLY. Well, GLY, we're learning, stands for glycine. And you can see that its functional group is simply an H. It is the only achiral center in our amino acids. And of course, the last one is PHE. Remember what it stands for? Phenylalanine. Phenylalanine has a CH2 with an aromatic ring for its functional group R. Those have to be memorized to have success in this part of our section. We know that. So we're asking to put those together as we create our amino acid. This would be a tripeptide chain as you're drawing those. So when we're looking at ILE, GLY, PHE, please pause your video and draw that into your notepad and come back when it's accomplished. Draw first, then come back. Okay, so now you've come back and you have found that you've drawn a tripeptide bond. Here is a peptide bond between the first amino acid and the second peptide bond between the second and third amino acid. We called ILE, remember how we're just highlighting, ILE would be everything I color code here in green. ILE, so then the GLY, glycine, is everything I'm going to color code in pink. And the final amino acid, PHE, phenylalanine, that's everything that I've color coded in yellow. So 3 separate amino acids. When we hydrolyze, we break the peptide bonds. Let me just color code those a little differently so they stand out. Let's color code those, let's say, with a nice blue color. So here's the peptide bonds, just in a different color now. We're going to add water across those bonds. So this is to hydrolyze. And all we now need to do, hydrolyze. We're going to add the water back in so that the functional groups form a Zwitter ion. The N-terminal ILE amino acid is a Zwitter ion, has the NH3 plus and a COO negative. That's the Zwitter ion for ILE. Secondly, we had the GLY. Notice again, you're forming the Zwitter ion, where at the N is protonated, NH3 plus, and the acid is deprotonated, COO negative. That's the Zwitter ion form of GLY, glycine. And phenylalanine is the same, where you're going to draw the NH3 plus, COO negative, Zwitter ion form of PHE, phenylalanine. So really the reaction itself is quite simplistic. It's another opportunity to keep practicing your 3-letter codes, making sure that when you're given those 3-letter codes, you can draw the amino acids given the tool page. You're going to break the peptide bond and just reform the Zwitter ion, the process of hydrolysis. I want you to practice some more. See how well you can do without looking up the 3-letter code structure, in terms of can you find, without cheating, what the ALA stands for without being told, the LEU and the GLY. What do those stand for? Go to your test-taking tool page and see if you can find those without relying on looking up the code. You're working to remember, I'm just going to go ahead and say memorize the code for each name. And once you know the name, you can use your chart to go through and find the structure of the amino acid. Alrighty. So just to emphasize, ALA, when you go into your code, ALA, I'm asking you to just remember ALA is associated with alanine, and that then you can look up that its functional group is CH3. Alrighty. LEU, how are you doing with that? What does it stand for? Well, I need you to know that LEU stands for leucine. Once you know leucine, you can easily find its R group, CH2CH, CH3 taken twice. But I have to know what LEU stands for so that I can find leucine on the table. And then, of course, GLY, glycine. That's the simplest one. That gives us the H for the functional group. SER. How are you doing with that? What does that stand for? Here it is, serine. CH2OH is its R group. The next one, THR, threonine. Here's its functional group, CHOH, CH3. I have to know that THR stands for threonine. And finally, PHE, which we looked up a little bit ago, for phenylalanine, phenylalanine. So that's what I'm asking. You memorize the abbreviations so that you can use your chart to find the functional groups. Draw those for me. Come back when ready. All right. Well, so you have drawn those. and you have separated those into each individual Zwitter ion. So here is the alanine, ALA, functional group. Here is the LEU leucines, functional group. And of course, glycine, the simplest of all, would have just a simple hydrogen. Notice that we hydrolyzed to form 3 separate Zwitter ions of amino acids. You next drew serine, CH2OH. The next one that came was threonine, CHOH, CH3. And of course, with phenylalanine, its functional group given here. The Zwitter ions for each were formed through the process of hydrolysis. The skill isn't tough, it's just another opportunity to practice using those charts. All right, good work. So denaturation is the process of altering the shape of a protein without breaking the amide bonds. So denaturation, we're not separating individual amino acids. But rather, you're allowing intermolecular attractions such as hydrogen bonding and van der Waals forces. All of those things are going to be disrupted. Can I write intermolecular forces as IMF and then remind you that those are the things such as hydrogen bonding, van der Waals forces, those sorts of things. So notice that we have a nice coiled globular protein in this structure here, and you would know that it's being held in position by what we would refer to as intermolecular forces known as hydrogen bonds. When you denature a protein, we're not separating out the individual amino acids, but rather you're breaking those intermolecular attractions so that it just simply uncoils and loses its three-dimensional shape. Now that can happen by a variety of ways, such as really high temperatures can denature a protein. Acidic and basic solutions can denature a protein. And even agitation can disrupt these non-covalent interactions that hold specific molecules in shape. Those are what we refer to as the intermolecular forces, which would include hydrogen bonding and those dispersion forces, and even the electrostatic attractions can be disrupted. Denaturation often makes the globular proteins less water soluble. They're all becoming, in sense, more non-polar. And let me give you an example. Ovalbumin, which is the major protein in an egg white, it's denatured when you cook it. It's denatured when you whip it to make egg white stiff. If you've ever made a lemon meringue pie, I have not, but if you have seen it done, or have done it yourself, you know that by whipping with a whisk egg whites, they'll become stiff. You're denaturing the proteins, turning them from a clear, colorless to an opaque and white color. So they lose their translucent nature when they become denatured. Let's talk about enzymes a little further. Enzymes, we know, are called nature's catalyst. Enzymes are proteins that serve as biological catalysts. And what the catalyst does, we've learned, is just speeds up a chemical reaction. It's the kinetics that are affected, right? It speeds up the rate. It can speed up the rate of a reaction up to 1,000,000 or 100 billion times faster. So really it turns a reaction that has no chance of progression without the use of a catalyst. Alrighty, so they are very important in running the kinetics of our bodies, of our biochemical pathways. Enzymes are very specific to what proteins they can act upon. So they are specific based on shape. Each enzyme catalyzes a certain reaction or one type of reaction only. So enzymes are very specific to their substrate. You'll notice that enzymes have a very specific naming system because they'll always end with the same suffix, ase. Look at these. Peptidase. Here the ase. Lipase, ase. Hydrolysis, ase. You'll know that this is an enzyme because you hear the ending ase. Sometimes enzymes require a cofactor. It can be a metal ion or an organic molecule to help the reaction secure itself to the substrate. So they're a necessary molecule or a metal ion that it serves really as a site of attachment for the reaction to occur. So as an enzyme and its substrate latch onto each other, the cofactor helps with that process. Let me give an example of one of these. And again, perhaps from your biology days and your cellular biology especially, these might be familiar terms. There is a molecule, NAD carries a +1. NAD plus is a cofactor. Sometimes you'll hear cofactors used as the word coenzyme. And it functions to oxidize lactate to pyruvate with the aid of a very specific enzyme called lactate dehydrogenase. Hear the name? ASE. Dehydrogenase. That's the name of the enzyme that's specific to lactate. Now, lactate, here's this molecule here. And notice that NAD plus is part of this reaction. It is needed for this reaction to occur. The enzyme sits on the arrow. It is not a reactant, it's not a product, it's a catalyst. All catalysts sit on the arrow. Remember that a catalyst, or an enzyme, same word, but an enzyme is in a natural biological pathway. They're not a reactant, they're not a product, they're there to speed the reaction along. So therefore, they're consumed in the first step, but regenerated in the last step to be used over and over and over again. The coenzyme is NAD+. It is a reactant. It is a necessary part of this structure so that the lactate dehydrogenase, the enzyme, can actually adhere correctly to its substrate, lactate. And when it did so, notice that it was reduced. We removed hydrogen ions. And so, it was oxidized. I said that wrong. It oxidizes by taking the hydrogens and putting them onto NAD and removing the acidic proton. So again, a cofactor is a molecule necessary to drive the reaction. It helps to bind the enzyme to its substrate. We are going to explore 6 categories of enzymes, and you must know these by name. And we'll take our time, but I'm playing it out here. You must know the 6 categories of enzymes and their function. We'll go through them one at a time. So these 6 categories by type of reaction the enzymes catalyze. The first of the 6, oxidoreductases. What do you hear in that word? Oxidoreductases. Hear the ASE, that means enzyme, doesn't it? Oxidation reduction. Oxidoreductases catalyze redox reactions. So when a substrate is oxidized or reduced, a coenzyme is required, which serves as the oxidizing or reducing agent. Just as we had said before, NADH here is serving as the oxidizing and reducing agent in this redox, which is transfer of hydrogen. Notice here that we have a COO and a carbonyl carbon here. In the presence of this enzyme, a dehydrogenase, you're going to add on, let's say this, we're going to increase the number of C to H bonds and you decreased the number of C to O bonds. Isn't that exactly what happened? Here we had a C double bond O. Now we only have one bond to an oxygen and more hydrogen added on. This is known as reduction. To oxidize means to increase the number of oxygen carbon bonds. To reduce means to reduce the number of carbon to oxygen bonds. This is an example of a reduction. Therefore, the type of enzyme that lactase dehydrogenase, it would be called an oxidoreductase enzyme. That would be the category it falls under. All right, the first of 6 is responsible for catalyzing oxidation and reduction reactions. Let's look at the second one. The second is a transferase. Transferase. What does that word mean for you? Well, it transfers a group from one molecule to another. So let's look at an example of a type of transferase. In the first example, here I have an alanine functional group. Alanine NH3+. It has a methyl group, a carboxylic acid group, and an H. And we have an alpha ketoglutarate molecule. On the products, and remember, you're not predicting what happens. You're just trying to classify what type of enzyme is responsible for this reaction. So what has happened over here? You notice that on one of these molecules, we had the NH3 group, and on the second one, we had the CH, or the carbonyl, C double bond O. What happened on the other side? They transferred, didn't they? Now, originally on the CH3C CO2 group, you now see that it has received the carbonyl group, C double bond O, and the NH3 group ended up in the larger molecule. These two functional groups transferred. Right? So it's just saying these guys switched places. And when they did so, the type of enzyme that regulates this metabolic pathway is known as a transferase, transferring one functional group for another between two molecules. Let's look at the third type of 6 called a hydrolase. What word comes to mind there? I think of water, don't you? Adding water across bonds. Hydrolase. These catalyze hydrolysis reactions, the cleavage of bonds with water. So here's an example of an enzyme. See how it's ending in ASE. What is its category? What does lipase fall under for category? Well, let's look to see what is the difference between the left and the right side. Well, right away I notice that I'm breaking bonds, right? Because I'm noticing that I have this backbone in a triaglycerol molecule, which we know to be one of the fatty acids from our previous chapters. And I can see that that backbone still exists over here, but what's been added on? We have these H's, don't we? And on the opposite side, we had this C double bond O attached to some functional group. And do you see how now all of a sudden we have an OH over here? We've broken bonds and added water across those bonds. When those bonds were broken, we added water across the bonds. It took 3 water molecules because we had 3 spots for it to go, right? 3 waters, 1, 2, 3 H's, 3 OH's. So it was necessary to break 3 bonds, each one of those requiring a water molecule. So we talked about hydrolysis, regulating hydrolysis reactions. Enzyme number 4 are called isomerases. Isomers, hear that root word, isomer? Isomerases catalyze the conversion of one isomer to another. All we're doing is rearranging the molecules. Rearrangement within the same molecule. So notice here, for example, we have an opportunity to see a CO, the double bond here, and then we have a CH2OH at the end of that molecule. What's changed from the left to the right side is really just the position where we find the CH2OH is now present here, and the C double bond O is now present there. It's just simply rearranged, forming an isomer. So in other words, they're the same number of each kind of atom, they're just connected in a different order. So if you just simply see a molecule rearrange, the type of enzyme responsible for that is called an isomerase. How about in number 5? Lyases. Lyase. Those are enzymes that catalyze the addition or the elimination to either add or subtract a double bond. That's the key of this whole definition. If you're noticing the addition or an elimination of a double bond, think lyase. So notice right here we have a double bond in the structure. Adding water across of it eliminated a double bond. When you eliminate a double bond, it's known as a lyase. So addition of a molecule to a double bond or the elimination of a molecule to give a double bond are happening through the enzymes known as lyases. Notice that in any one of these structures, to categorize this enzyme, you need a double bond. All right. The 6th and final category of enzymes are called ligases. Hear that hard G, ligases. These catalyze bond formations by energy releasing from a hydrolysis reaction. It's energetically unfavorable, so that energy released in a hydrolysis reaction is used to drive the reaction. So when I see an ATP, that's a high energy molecule called adenine triphosphate. ATP molecules, high energy. And it goes into an ADP, you know that it's spent one of its high phosphate bond energies. So that's a release of energy. Here we have pyruvate plus carbon dioxide. And on the other side, notice that we've added on the carbon dioxide to the molecule, that the carbon dioxide was adding on. So catalyze bond formation means to lengthen the molecule. A bond forming is going to increase the length of the overall molecule. We added carbon dioxide to the original molecule, increasing the length of the molecule, and it cost energy to do that, to form new bonds. All of those types of reactions would fall under a big category of enzymes known as ligases. When bond formation accompanied by energy release through the process of hydrolysis. Here is a table that you should really spend time and get to know very well. When I say get to know it, we need to know the categories of these 6 enzymes. We have presented them one at a time. We said the first of the 6 is an oxidoreductase. Oxidoreductase. Think redox. And underneath that, you can see that we have specific subcategories of oxidoreductases. Oxidation, reduction, an additional removal of 2 hydrogens. Oxidation is taking care of oxidases. Reduction happens by reductases. And dehydrogenases add or remove hydrogens. To oxidize increases the number of CO bonds or decreases the number of CH bonds. To reduce increases CH bonds and decreases CO bonds. When you see that happening, you're under the category of oxidoreductases. The second, transferases. Underneath there, we see 2 subcategories. Transaminases transfers the amine group, and you can have a kinase, which transfers a phosphate group. We'll need to know the major category as a transferase. The third word to know, hydrolase, to hydrolyze. To hydrolyze means add water across the bonds, and you're ending up breaking bonds. We hydrolyze the amide bonds to separate proteins into their building blocks, amino acids. The fourth category, isomerase, isomerization. That's really just a rearrangement of the same atoms in the same molecule. A lyase, L-Y-A-S-E, a lyase was the 5th category. Think about the double bond in this category here, to add or remove a double bond. A dehydrase removes water, a decarboxylase removes carbon dioxide, and a synthase adds a small molecule to the double bond. Those are the subcategories under the 5th that we'll need to know, lyase. And of course, the 6th was a ligase. Hear the hard G in there, ligase. This is the bond formation accompanied by ATP hydrolysis, the bond formation between a substrate and a carbon dioxide. You're going to add carbon dioxide and spend energy. So you'll literally see ATP turning into ADP as another clue of that type of enzyme. Know this chart. Know it backwards and forwards. Know the chart so that you can categorize types of enzymes. Let me give you an example. What type of enzyme is used in the following reaction? We have to tell what category of an enzyme. So the first thing I'd recommend is figuring out what the heck is happening from the left to the right-hand side. Alrighty. So I noticed that, you spend some time observing, that I have the same portion of the molecule on the left and the right-hand side, but what I see is that I have split out a carbon dioxide. So I broke a bond right here. A bond broke. Now, what type of activity is going to break a bond and release a carbon dioxide? Can you tell? Until you get familiar, go back into your chart, and what do you see? Where do we break bonds? Giving you think time. Can you find it? I hope you're noticing that when we found what type of bond it was, did you see the removal of a carbon dioxide? What are you thinking? The reduction involved the loss of a molecule, carbon dioxide. It formed a double bond. Now you might not see that, but carbon dioxide, C double bond O. So the enzyme is a lyase. That's the answer we needed to hear. Because carbon dioxide was removed, it was subclassified as a decarboxylase. Did you see that? A decarboxylase. We had highlighted that right here. It was a lyase, subcategory decarboxylase. It removed a carbon dioxide. Yep, we got to know that. It takes practice, doesn't it? How about another? What's going on in this picture? Well, again, I just want to emphasize it's going to take some time to figure out and recognize patterns. Some of the things I notice is that I'm adding water. Do you see that? How I'm adding H2O? And I'm breaking a bond. This bond broke. That bond separates this structure and this structure. It's called a peptide bond, isn't it? We added water and broke a bond. As a matter of fact, we broke an amide bond, didn't we? So what does that fall under? We got to practice. Go into your chart and study what that looks like. Do you notice? We added water and broke bonds. So that brings us right down here, doesn't it? A hydrolase. And specifically, we broke the amide bond of an amino acid group, didn't we? So in this example, as we progress through and practice, we're noticing that the reaction was hydrolysis. That was the cleavage of a bond with water, so we know it was a hydrolase. And because it cleaved an amide bond in a dipeptide, we classified it as a protease underneath that category. So in that example, we were practicing here, as I just kind of slide again, we said we broke the bond, so it was a protease and a hydrolysis, adding water that broke a bond, the bond that was an amide bond between two amino acids, so the main category of a hydrolase with a subcategory of a protease, the third of the 6 types of amino acids. How about these two? I'd really recommend you pause the video, practice these two with your chart so much that you get familiar with how to read that chart. It really just takes an incredible amount of practice to get those enzymes memorized. Pause, come back when ready. Okay, you must be back. Well, look what I found when you studied this molecule. Do you see something about a double bond? And when I notice the double bond, and I see that I'm kind of removing an HOH to form a double bond, what is that enzyme that we thought about with double bonds? I highlighted it here, didn't I? That a lyase, L-Y-A-S-E, a lyase is adding or eliminating a double bond. And we said specifically this one removed an HOH, so it was a dehydrase. In that example, the first was a dehydrase. And we had that recognized here. How about in the second example? What did you notice here? You got to really study the picture. I noticed that there's a group of PO3 2 negative on the right side of the molecule and an OH group down here. What happened from the left to the right? What happened? They just switched places, didn't they? Nothing more than a rearrangement of functional groups within the same molecule. We said that this one removed a water and formed a double bond. This one is a rearrangement. Now, what type of enzyme undergoes rearrangement? That's not very neat. And I'm sure you went through, just to kind of go back, you're studying this chart, the rearrangements. It's one of the least talked about in here, isn't it? It's isomerization is what that was. Just a rearrangement of functional groups to create a constitutional isomer. So you named that an isomerase. And that's the skill here, really, is recognizing the pattern of change. When two functional groups switch places within the same molecule, you get an isomerase category. Excellent. Keep practicing those. Go back and find many, many examples to keep practicing in your text. So just reminding you how enzymes work as we kind of bring closure to the lesson. An enzyme contains an active site that binds to the substrate, forming an enzyme substrate complex. So here's the active site. It really is by unique shape. An enzyme with an active site fits the substrate, and you form an active substrate where you have an active site that's bonded here. So the enzyme substrate complex, and there's really two competing theories how this occurs. One of them is called the lock and key theory, and it really just is the first of two based on the specificity of the substrate of the enzyme's active site. So if I have, the enzyme substrate complex releases the two products, the enzyme is released in the original form, and it's used over and over and over. And those two competing theories, the first of the two is a lock and key theory. That model simply states that the active site is rigid and that must have an exact complementary shape to the substrate. So just as you have a whole ring full of keys, but only one fits the lock. The lock and key theory. You have a whole set of keys, but only one of those keys is going to fit the lock to actually open the door, for instance. So it's a very rigid state that this active site in its three-dimensional shape is complementary to the substrate, and only this substrate will fit in there. Any other substrate will not fit in, and therefore the reaction would not occur. Now, a second theory is called the induced fit model, which says it's a little more flexible in shape, that I can kind of go in and it can mold to the shape of the enzymes. And so really the scientists are kind of competing back and forth. I know that the lock and key theory is more acceptable, more widely used in describing how enzymes work, that it's a very specific way that enzymes fit their substrates. But just so you know, there is an induced fit model theory that competes with that as well. So just that shape of the active site before binding, the shape of the active site after binding, and this shape just kind of molds itself to the enzyme for the reaction to occur. The activity of an enzyme is affected both by the temperature and the pH. And we can see an increase in temperature increases the rate of most chemical reactions. And so enzyme catalyzed reactions exhibit an increase in rate. And you can see that optimally about 37 degrees Celsius, which is body temperature, 98.6 degrees Fahrenheit, tends to be the optimum temperature for enzymes to run their reactions. So at this point, you've finished out your note pack with me. You have some assigned reading to make sure you've completed. And when ready, go ahead and submit this note pack. It's been a pleasure working with you for Chapter 21. Hey friends, just one more chapter. We're in the home stretch.